Okay, Assalamualaikum and greetings everyone. I am going to continue on lecture on the nervous system. Okay, um, I am not going to like focus every part of it. Okay, but I am going to focus on especially on the action potential. Okay, because the rest of it and the rest subtopic of this lecture, I believe that it's not really that complicated or hard to understand. Okay, so some of the subtopics that I think is not really that I'm not gonna say important, but it's easy for you to understand without me explaining it. Okay, so okay, this is just an example. Okay, just intro. Okay, it's a tropical cone snail. Okay, they are small, they are slow moving, but yeah, oh, they are dangerous hunter. They are carnivorous. They're gonna hunt. They're gonna kill. They can devour fish. Okay, can you imagine that? And it able to paralyze their free swimming prey by injecting venom. Okay, with the harpoon-like tooth over here. So the venom is so deadly, unlucky scuba divers have died from a single injection from this tropical cone snail. Okay, so what makes it so fast at acting and lethal? How? Nampak macam slow. Well, how is it so fast acting? Okay, I mean like how is it? How can it make the scuba divers die due to the venom? Okay, it's actually there's a mixture of toxic molecule. Each has a specific mechanism of disabling your neuron. Okay, there's a certain kind of chemical that is going to stop your neuron from functioning. Okay, so that's just an intro lah. Okay, we have these are the nervous system of different kind of organisms. Okay, the individual neurons, which is the purple organized in a diffused net. Okay, more sophisticated nervous system, especially for the higher level of organisms, especially we human or other multicellular organisms. Okay, is gonna have neuron, okay, nerves and the ganglia and also the brain. Okay, you see the Nidirian or the Hydra doesn't have any brain, nerves or neuron. Okay, they are quite simple. All right. I believe you guys um do know, I mean like know the differences between the CNS ataupun the central nervous system and the peripheral nervous system. Okay, the central nervous system consists of your brain and also the spinal cord. Okay, is one of the yellow part over here. And the PNS, okay, consists of left, right pairs of cranial nerves, spinal nerves, ganglia. Okay, so that is the CNS and PNS. Okay, the same thing. I'm gonna skip this one. Okay, this is the organization of vertebrate nervous system. Okay, we have the gray matter. All right, is made of neuron cell bodies. Okay, and we have white matter over here, consists of a bundled axons, learning, emotion, processing sensory information, generating command, everything happen in the white matter. Okay, and we have ventricles over here as well. Okay, in the middle. All right. Want to skip this one as well. Okay. All right. So the human brain, this is where, this is the structure of the human brain. Okay, our brain is enclosed in a skull. Okay, and we have cerebellum over here. Okay, and that controls your what you call the body balancing. Okay, the medulla oblongata, where some of their let's say that the medulla oblongata is controlling your involuntary action of your body. Okay, we have the spinal cord over here. Okay, which connecting the brain to the other peripheral nervous system. All right, and we have the cerebrum, which is the largest region of your brain, controlling your emotion, memory, feelings, everything is at the cerebrum. You have the thalamus, the hypothalamus. The hypothalamus normally we associate them with the homeostasis, and we have the pituitary gland, the anterior and the posterior. All right. So this is the same thing, the frontal lobe, okay, where it involving the decision making, planning, everything, okay, and then we have the parietal lobe over here, okay, more on the sensory association of cortex, okay, and then the we have the visual association cortex over here as well, okay, occipital lobe, okay, these are the okay, and we have the temporal lobe as well over here, okay. I think this is a more detailed explanation of the human brain, okay, and all right. Okay, now we are going to look at the neuron structure and function. Okay, so this one, okay, this is how the neuron looks like. We have the nucleus, okay, and then this is the cell body. All right. So most of the neurons organelle, including the nucleus, is located inside the cell body, and then the cell body is studied, or they have a projection ataupun like finger-like projection like that, okay, with numerous highly branched extension called as a dendrite. It's a like hair structure, dendrite, okay, projecting out from the cell body. Together with the cell body, dendrite is going to receive signal from other neurons. So this is where they are going to receive the signals. Okay, and then connecting the cell body to the dendrite over here, okay, is the we call it as an axon, an extension that transmits the signals to another neuron, okay, longer than the dendrite. Okay, dendrite is shorter, but axon is normally way longer, over a meter long for giraffe to reach spinal cord. Okay, and then we have synapse over here. Where is synapse? This is synapse. Okay, you can, if you zoom in, meaning the end. Okay, over here, the end of the what do you call it? The synapse over here, connecting to another neuron. Okay, that is where we are going to learn that one later lah. Okay, each branched end of an axon transmit information to another cell at a junction. Okay, where it's going to involve some chemicals and action potential over here. And what is a neurotransmitter? Okay, at most synapse, chemical messengers called as neurotransmitter is going to pass. They're going to pass the chemicals, the substances from the transmitting neuron to the receiving cell. This is the receiving, this is the transmitting. Okay. Okay, the transmitting neuron we call as a presynaptic cell. The one that is going to receive we call as a postsynaptic cell. Okay, it can be neuron, it can be another neuron, it can be muscle. Okay, once the muscle receives the signal, they're going to contract, or it can be a gland cell, meaning when the gland has received signal from the presynaptic cells, is going to be triggered and release hormone. So there are three possibility over here. Receiving the signal as the postsynaptic cell. Okay, so this is the basic morphology of the neuron. We have the dendrite is going to receive the signal. We have the axon hillock, okay, like a neck of the body cell, cell body, and that is where the action potential is going to be generated. What is an action potential? We're going to look at that one later. We have the axon over here is going to transmit the signal from the cell body to another cell. And we have the synaptic terminal, and the terminal over here, this one, where the neurotransmitter is going to be released. Okay, meaning from electrical impulse into chemical impulse. Okay, the chemical messenger pass information to the postsynaptic cells. Okay, so the neurons of vertebrates and invertebrate require supporting cells called as a glial cells ataupun called as a glia. Okay, the function is more on nourishing the neurons, protecting the axons, okay, and then regulate extracellular fluid surrounding the neuron and replenish the group of neuron in transmitting information. All right, so just basically it's more on nourishing, providing all the nutrients needed by the neuron, insulate ataupun act as an insulator of the axon. Okay, now we're going into the information processing. Okay, we have information processing by your nervous system is going to occur in 3 different stages. We have the sensory input. Okay, that should be the first one, where it's going to transmit information about the external stimuli. It can be light. You nampak light je, you're going to like, you can see something. Someone touches you, that is a stimuli as well. Okay, you're going to turn around because the touch makes you turn around. That's the stimuli. Or internal condition can also be the stimuli. It doesn't necessarily be an external stimuli. Let's say there's a difference ataupun there's an increase of your blood pressure, ataupun there's a muscle tension happening in your body. Okay, that can be considered as a stimuli ataupun sensory input. And we have the integration, meaning what's happening inside the what happened after your stimuli is generated. Okay, the local circuits connecting neuron in the brain or ganglia are responsible to for analysis and interpret of the sensory input. Okay, integration. So it's going to integrate, it's going to interpret whatever stimulus that you have received. Everything is going to happen in your brain. Okay, and then once your brain has interpret all the information received by your brain, and then they're gonna create an order or information ataupun to the motor output. Okay, it's going to transmit the signals to the muscle cells, causing them to contract. Okay, the brain asks the muscle to contract because after analyzing all the information received from the sensory input ataupun from the stimuli. Okay, at the same, not necessarily muscle, okay, meaning you have to remember motor output can be 2. It's of the muscle cells, okay, you're gonna cause your body to move, okay, your muscle to move by contracting and relaxing. Or it can be triggering a gland activity ataupun any gland that is going to release the hormone. And there are 2 possibilities for the motor output. Okay. We're going to skip this one as well. Okay, so this one, we have a stimulus over here. Okay, should be the stimulus where you are tapping the tendon connecting to the quadricep of your muscle. Okay, and then the sensor, okay, the sensor or the receptor is going to detect the stretch in the quadricep. So the sensory neuron is going to send the information or the stimulus to your spinal cord. Okay, so the cell body, this is the cell body of sensory neuron in the dorsal root ganglion. So once it reaches your what do you call your spinal cord over here, okay, there will be there will be a gray matter over here, okay, where you can find the end of the what do you call it, the sensory neuron, and there's an interneuron connecting the sensory neuron and also the motor neuron. Okay, so the motor neuron send the signals to your quadriceps, okay, causing it to contract and making your leg to jerk forward ataupun make your straightening your leg. And the interneuron over here, which is located in the spinal cord, also receiving the signal from the sensory neuron. Okay, so what can you see over here? Everything happens without involving your brain. Okay, not everything is going to involve your brain. Okay, this action that is making your knee jerk punya process over here doesn't involve your brain, but it is involve your spinal cord. Okay, which still involving your central nervous system, right? Okay, because this kind of action or this kind of process doesn't involve your brain because it's just what do you call that? Is a really quick action. Okay, doesn't need the brain to interpret whatever information is received. Okay, and this is the diversity of neuron, meaning we have the sensory neuron. This is how it looks like. Okay, the cell body is located at the axon. Okay, we have the interneuron is quite branchy over here. Okay, and the motor neuron is like a normal neuron that we learned before. Okay, the normal structure of a neuron. So depending on the role, okay, in information processing, the shape of neuron can vary from simple to complex one. Neuron have highly branched dendrite, can receive input through thousands of synapses. So neuron that transmit information to many target cells also have highly branched axon. Okay, so depending on their role, okay, in transmitting the information. Okay, now we are looking at ion pumps and ion channel. Whatever happened inside the neuron, we're going to go into detail. Okay, so it is going to involve a lot of ion channels, ion pumps, to transmit to create an action potential to depending on the strength of the stimulus. Okay, so in neurons, okay, ions are unequally distributed between interior cells, meaning inside the neuron, and surrounding fluid outside of the neuron. They are unequally. It's not going to be equal ions ataupun charge between positive and negative. As a result, inside of the cell is going to be negatively charged. Always remember, it's going to start off with negatively charged inside the neuron. Okay, this charge difference, or we call it as a voltage, okay, across plasma membrane is called as a membrane potential. Okay, membrane potential is going to be created when there's a difference of positive and negative charge between inside of the neuron and outside of the neuron. So during resting potential, during resting potential, the neuron is resting, meaning there's no any signal, there's no any stimulus is sent. So normally the charge is going to be negative 60 to negative 80 millivolt. Okay, so it's quite negative because, you see, because the inside cell is negatively charged. Okay, next one is the formation of the resting potential. How are you going to create the resting potential of the neuron? Okay, so over here, the potassium and sodium plays a really important role in resting potential. Always remember resting potential, meaning there's no any stimulus is received by the neuron. Okay, so they have concentration gradient across plasma membrane of a neuron because there's a difference in terms of charge between inside and outside. So there's a difference. Okay, they have a concentration gradient. Maknanya dalam dengan kat luar tu tak sama dia punya charge dia tu. Okay, most of the neuron, the concentration of the potassium is going to be higher inside the cell, whereas the sodium is going to be higher outside. Sodium tinggi kat luar, potassium tinggi kat dalam. So the potassium and sodium gradient is maintained by sodium potassium pump, or we call it as a SPP. Okay, but make sure during exam don't write the SPP lah. Okay, tulis penuh. Alright, so what does it do? It's going to transport 3 sodium out of the cell. Okay, but at the same time, 2 potassium ion is going to be transported inside the cell. Okay, to maintain the resting potential. So the pumping generate a net export of positive charge. Okay, just a few millivolt, and it's going to act quite slowly lah. Because you have to remember the ion channel, the opening and closing is quite slow. It happens quite slow actually. Okay. All right, that is okay. So remember, tadi tu, the potassium is going to be higher inside, sodium is going to be higher outside during resting. During there's no any stimulus, tak ada apa-apa yang menyebabkan awak triggered in your body. Okay, so let's see the concentration inside and outside of a mammalian neuron. Okay, the intracellular, using the potassium is going to be way higher. Okay, and the sodium is going to be less, but extracellular, meaning outside of the neuron, they're going to be really high. Okay, the same thing applies to the chloride ion as well. Okay, but we are focusing on these two only for now. Okay, so again, this is during the resting potential. Tak ada action potential, tak ada stimulus datang lagi. So ion channels allow ions to diffuse in and out across a membrane. Yeah, true, right? Okay, that is the function of the ion channel, allowing the ions to flow in and flow out. When ions diffuse through channels, they're going to carry units of electrical charge, kan? Dia bawa ion, meaning if you are allowing the sodium ion to flow out, okay, more sodium, more positively charged is going to flow out. You're going to make it more negative. That is what it means. They carry unit of electrical charge. There's a leaky channel over during the resting potential as well. Okay, the diffusion of potassium via potassium channel that always open. Because we want to make it more negatively charged during resting potential kan. So the potassium channel is going to be always open to make sure that all of the potassium ion always flow out. So you have to remember when there's a question asking like what is happening during resting potential, you're going to see that a lot of potassium channels is going to open. Because they want to make sure all the potassium ion, nampak tak? The diamond shape ni, okay, to flow out, to make sure that it is more negatively over here, because all the positive dah keluar, jadi negatif lah kat dalam. Make sense, right? Okay, but just a few sodium channel is gonna open. Sodium and other ion cannot readily cross the membrane, okay, because just a few sodium ion channels open, so but they cannot diffuse through the phospholipid bilayer lah. So making a lot of sodium ion is accumulated inside the axon. Potassium outflow leaves a negatively charged inside the cells. As I mentioned before, when there's a lot of potassium ion flow out, they're bringing positive ion flow out. So when there's a lot of positive ion flow out of the neuron, you're creating a negatively charged. Sebab tu semua negatif kat sini. Okay, so the build-up of negative charge inside the neuron is the major source of membrane potential. And you are creating a membrane potential because there's a difference between charge inside of the cell and outside of the cell. You are creating a potential. You cannot make it the same. If the charge is same between inside and outside, there you're not creating any potential. You're not allowing the action potential to happen. Okay. So yeah, see the difference. The potassium channel allowing only potassium to flow out, sodium channel allowing sodium to flow in. Okay, but the sodium potassium pump tu yang that the one that says that allowing 3 sodium flows out, 2 potassium flow in. All right, so membrane potential is a differences in the concentration of ion on the opposite side, meaning between inside of the neuron and outside of the neuron, okay, leading to a voltage called the membrane potential. Okay, so this is the basis of membrane potential. So we have, the same thing lah. The purple one over here generates and maintain the ionic gradient of sodium and potassium. Okay, it plays a important role, especially during the resting potential. And we have the potassium channel over here. Many potassiums are open, a big outflow of potassium. These play a major role, especially during the resting potential as well, because they want to make sure a lot of potassium to flow out. Okay, and only a few are open, only little diffusion of sodium. Okay, near to none. Okay, during the resting potential. All right. So this is the modeling. I'm going to skip this one as well. All right, so action potentials are the signals conducted by axon. Okay, how does a stimulus bring out change in the membrane potential? Meaning, when there's a stimulus, okay, when your retina receives light, or when someone touches you, when someone anything, okay, or ataupun when your blood pressure increased, so those are considered as a stimulus. Okay, some ion channels in a neuron are gated ion channel, right? Meaning gated ion channel, meaning ion channels that is going to open or close depending to a stimulus. So when a gated ion channel tu yang sodium tu lah, sodium and potassium channel tu, whether it open or close, it changes the membrane permeability to the particular ion. Yalah, when you're talking about sodium channel, you're only allowing the sodium ion to flow in, right? Only particular ion, not the same, not all ion is allowed to diffuse in and out. So this is going to change in permeability, allow a rapid flow of ion across the membrane, changing the membrane potential. This is the ion channel. This is the ion. Okay, it is closed. Okay, no ions is going to flow across the membrane. Okay, but when there's a change in membrane potential, let's say there's a stimulus. So the stimulus is going to make sure you want to make the ion channel to open, allowing the ion, okay, from the extracellular to flow into the cell or to put into the neuron. Okay, so we have a hyperpolarization and depolarization. If we get it, potassium channel in a resting neuron is going to open, the membrane permeability of a potassium is going to increase. Okay, when the potassium channel open, they are going to allow the potassium to flow, and then it allowing the potassium ion to flow in. So that's why it says increasing the permeability of the potassium. So as a result, the net diffusion of potassium out of the neuron is going to be negative 90. It's going to be even more negative than the resting potential that again. That is a hyperpolarization, increase in magnitude of membrane potential, making the inside of the membrane even more negative than the resting potential. Opening some other types of ion channel has opposite effect, making the inside membrane is going to be less negative. Tadi more negative, now it's less negative or more positive. So depolarization is reducing in magnitude of the membrane potential. So depolarization often involves gated sodium channel. Okay, so ingatlah, hyperpolarization when the potassium channel opens. Depolarization when the sodium channel opens. So let's say if a stimulus arrives, it's going to cause the sodium channel to start to open. Kan, tadi resting potential tu, the sodium channel, only a little sodium channel open, right? But now when the stimulus arrives, they're going to cause the sodium channel to open, a lot of them to open. So the membrane permeability to the sodium is going to increase, allowing a lot of sodium ion to flow in. So the sodium diffuse into the cell along the concentration gradient, causing depolarization. You see, it's going to the voltage is going to be positive 62 millivolt, whereas during the hyperpolarization is going to be negative 90. It should be millivolt lah, this one, sorry. Okay, so the response to hyperpolarization and depolarization is simply a shift in a membrane potential. It's a shifting. What does it mean? So the shift is actually a graded potential. Okay, the magnitude that varies with strength of stimulus. Okay, maknanya if you have a larger stimulus, Okay, the largest stimulus, meaning someone slapped you so hard, okay, is going to greater change in the membrane potential. Okay, so but the gradient potential is going to decay with time and distance from the source. It's going to decay, it's going to decrease with time and the distance. Maknanya bila dia makin jauh daripada source, it's going to decrease. That is called as a gradient potential. But action potential is actually the massive change in the membrane voltage. Maknanya, because we all of us understands that during the resting potential, inside of the neuron is going to be more negative, right? But when there's action potential, is going to make inside of the neuron more positively charged. So itu yang dikata massive change in the membrane voltage. They have a constant magnitude. Okay, can regenerate in adjacent region in membrane. Okay, you see, it's going to regenerate to the adjacent, maknanya bahagian tepi, along the axon. So it can spread along axon, well suited to transmit a signal over long distance. Okay, still under the graded potential, so some stimulus is going to trigger a hyperpolarization. Okay, remember the hyperpolarization is going to create a more negatively charged than the resting potential. Nampak tak ni? This is the resting potential. It's around -80 like that. But the hyperpolarization is even more negative. An increase in the magnitude of the membrane potential, the inside is going to be more negative than the outside. It's either caused by outflow of positive ion, maksudnya lebih banyak potassium ion flow out, ataupun there's an inflow of negative ion causing it to be more negative. But since we are talking in neuron, inside the neuron, because there's a lot of potassium ion outflow, and flow out from the neuron cells. So it's going to be even more negative than the resting potential. Okay, so why? What makes it like that? Because the larger stimulus produces a larger hyperpolarization. Now we look at the depolarization, a reduction in the magnitude of the membrane potential. So in the neuron, actually it happens when there's a sodium ion inflow ataupun sodium ion diffuse into your neuron. So in most neuron, the depolarization happen only up to a certain membrane voltage called as a threshold. Okay, this is a threshold over here. If you look at the dotted line over here, there's a threshold. Okay, so gradient depolarization produced by two stimuli that increase the membrane permeability to sodium. So the larger stimulus also is going to produce larger depolarization. Okay, you have to remember benda-benda ni semua berlaku at certain part of neuron, not all part. The hyperpolarization and depolarization at a certain part of the neuron. Okay, when there's a stimulus. Okay, so action potential, let's see. A stimulus is strong enough to produce a depolarization. Okay, depolarization making it more positive kan, maknanya dia akan makin menaik lah. It's going to increase, getting higher value. Okay, so it triggers a different type of response called as a action potential, and it's going to fire the neuron, meaning it's going to trigger lah the action potential. So you look at over here, this is the resting potential. Okay, over here, around -70, 80 like that. Okay, when there's a stimulus, okay, it's gonna depolarize. Dia dah naik, start naik slowly to depolarized. Okay, and then what makes it depolarized? Because of the sodium channel starts to open. So allowing all the sodium ion to rush inside the neuron. It's slowly increasing. If you wonder, kenapa dia macam slowly, why it doesn't like shoot up like this? You have to, as I told you before, okay, the sodium channel or the gated channel, the process of opening and closing is slow. So that's why the influx ataupun the increment of the value ataupun the voltage is slowly increased as well. okay, due to the slow opening of the sodium channel. And syaratnya, it must reach the threshold. The stimulus must be really high ataupun enough to reach the threshold. Kalau stimulus tu takat macam orang jentik je ataupun taklah jentik, still a stimulus. Someone touches you but is not that large. I mean the action potential is not that large ataupun really not significant. Again, maknanya dia sampai sini je, dia jatuh balik. It doesn't reach the threshold, so it's not going to create the action potential. Yeah, so that's why the threshold is important. Okay, so when the sodium channel open, all the sodium rush in, and it's going to increase steeply over here. It's going to increase really high, I mean really fast over here. Okay, and you have to remember when when the action potential starts to increase, okay, ataupun the membrane potential value starts to depolarize, okay, to become more positive. Okay, what do you say that this one over here, there will be even more sodium channel to open. It's going to make sure that all the sodium ion flows inside until it reaches the peak. Okay, the action potential reaches its peak, which is around maybe it depends actually. It's not going to reach 50. Okay, but maybe around 40 macam tu. Okay, but so once it reaches the peak, the sodium channel starts to close. Starts to close, but at the same time, this one sorry lah, the arrow should be over here, not this one. This is quite confusing, but you have to remember when the sodium channel start to close, after it reaches the peak, the potassium channel starts to open, making it more negative. Dia dah start nak jadikan dia negatif balik. So it's going to decrease. The membrane potential value is going to start to decrease. Okay, decrease over here. Remember this one we call as a hyperpolarization because you make it more negative. Okay, so until it's even more negative than the resting potential, nampak tak? You see why? Apa yang menyebabkan why it doesn't like stop over here and then it's going to rest return back to the resting potential? Because as I mentioned just now, okay, the closing and the opening of the potassium channel happens really slow. So sepatutnya bila dia dah reach the resting potential level over here, okay, the potassium channel, I mean the sodium, of course the sodium channel is already closed, okay, tapi the potassium channel starts to open, right? Tapi sepatutnya at when it reaches over here, some potassium channel should close as well, okay, to make sure that it reaches the supposed value of the resting potential. But since the closing happens too slow, so terlebih banyak pula potassium that flows out, making it even more negative than it should be, ataupun making it more negative than the resting potential. But no worries, okay, and the potassium channel will ataupun if you still remember during the resting potential, there are some of the sodium channels still open, right? A little sodium channel is open, so making it balance until it really reaches the resting potential, stabilizes it. That is why there's a hyperpolarization happen over here. So again, the same thing during resting state, the gated sodium and potassium channels are closed. Okay, but over here is already negatively charged. Inside and outside is more positive because of the more potassium ion has flow out. And then when the stimulus arrives at the neuron, okay, it starts to depolarize. So again, guys, if you are required to explain about the action potential arrives at the neuron, you have to make sure to explain the name of the process, resting, and then depolarize. What happened depolarize? Number 2 over here, it starts to increase. A stimulus is going to cause some of the sodium channel, okay, to open. and then making all the sodium ion to flow ataupun to diffuse inside the neuron is going to depolarize the membrane. So that's why you call it a depolarization. So until it reaches the threshold over here, okay, it's going to trigger an action potential. That's why it shoot up. Okay, so number 3, okay, depolarization opens most of the sodium channel. Okay, but the potassium channel is going to remain closed. Because you want to make sure it's going to keep on increase in the millivolt, and then the sodium influx makes the inside membrane more positive. Okay, number 3 though. So until it reaches the peak over here, okay, so bila dah sampai the action potential reaches its peak, the sodium channel start to close. Okay, terbalik pula, the potassium channel starts to open, allowing the potassium outflow, making it more negative balik. So the inside of the cell is going to be more negative again. And that is we call as a falling phase of the action potential. And then we have the refractory period. What is it? It's a downtime when the second action potential cannot be initiated due to inactivation of the sodium channel. Okay, meaning during this time, refractory period meaning when it goes down, okay, the sodium channel is already inactivated. Maknanya dia is not going to allow another action potential to happen over here. Maknanya kat sebelah ni there's another action potential. It's not going to happen. Because during refractory period, during the action potential is decreased, okay, it's going to make sure all the sodium channel is not open to allow another action potential to happen. Everything happens one by one. Okay, and then undershoot over here, which is the purple one, the one that I told you just now, sodium channel is going to close, okay, because you're only allowing potassium channel to open. But some of the potassium channel are still open. Why? As I said, the closing of the potassium channel happen quite slow. So terlebih pula potassium yang terkeluar, so making it more negative than the resting potential. So when this potassium channel close and sodium channels open, the membrane is going to return to the resting state. The resting state, remember, is aided ataupun helped by the sodium potassium pump, yang 3 sodium go out, 2 sodium potassium flows in, okay, making sure it maintained at the resting potential over here. Okay, so this is the conduction of the action potential. An action potential is generated when the sodium flow inward. The sodium flow inward, because remember, when there's an action potential, ingat je, the sodium is flowing in. Okay, making it more positive. Okay, because during resting potential tadi, dalam dia lebih negatif at one location only over here. But depolarization of the membrane is going to spread at the neighboring region, the adjacent ataupun the next region to it. They're going to spread to the next one, reinitiating the action potential there. So when the action potential is already complete, the membrane is going to repolarize as the potassium flow outward. Bila dia dah sampai at the next region over here, you see, the potassium is going to flow outward again. Why? To make sure it's back to the resting state. Action potential is going to move from one region to another. So bila dia dah lepas kat tempat yang, I mean like when the action potential reaches to the next region, the previous area is going to back to the resting state. Okay, the depolarization and repolarization process is repeated in the next region. Okay, the local current of ion across plasma membrane causing the action potential to be propagated along the axon. So nampak tak ni, and then go over here. The blue region is going to dah sampai sini, and then next is going to reach over here pula. So one by one. Okay, next one. This is the same thing lah. Basically, the same thing. As an action potential travels down the axon, there's a change in the polarity across the membrane. Because when the action potential arrives, you're going to make it more positive inside of the neuron. And the sodium and potassium gated ion channel is going to open and close as the membrane reaches the threshold potential. Must reach the threshold. Kalau tak, you're not creating any action potential. Okay, so at the beginning of the action potential, sodium channel open, allowing all the sodium to flow in, making it more positive, and sodium is going to move into the axon, causing depolarization. But what about repolarization? When the potassium channel starts to open, moving out the axon, making it more negative again. Inside the neuron is more negative because you want to make sure it back to the resting state. Creating a change in polarity between inside and outside of the neuron. Impulse is going to travel down the axon in one direction only to the axon terminal where it signals other neuron. So this is basically the Schwann cells and the myelin sheath. Okay, so the myelin sheath is covering the axon, act as an insulator. Okay, the electrical insulation that surrounds the vertebrate axon. So the same like your wire kat rumah lah. Kan, your wire, the copper is covered by a lot of different colors of insulator kan. Wire tu ada yang merah lah, biru lah, because act as an insulator. So the same thing applies to the neuron. They have an insulator as well, to reduce the energy loss, the heat loss and all. So the myelin sheath are produced by the oligodendrocytes in the central nervous system and the Schwann cells in the peripheral. Okay, there's a gap in the myelin sheath over here. Okay, we call it as a node of Ranvier. So the function is to allow the ions to diffuse in and out of the neuron. So this part yang berlakunya sodium flows in, potassium flows out, and not at the myelin sheath. So the propagating the electrical signal down the axon known as a saltatory conduction. Okay, so look at here. Even there's a node of Ranvier. So the node of Ranvier tu yang ada gated channel. So in a myelinated axon, okay, the depolarizing current during an action at one node of Ranvier spread along the interior. It's going to spread interiorly, happening inside of the neuron. Okay, so where the voltage gated sodium channel over here is going to allow the reinitiation. Okay, so maknanya when the action potential arrives over here, the sodium channel is going to open, making it more positive over here. Nampak tak? So maknanya during resting state is going to be more negative. So the action potential appears to jump from one node to another, from one to another. So it's going to travel really fast. Okay, instead of depending berlaku along the axon saja. So this is what happened when it reaches at the synapse, hujung neuron tu. So during resting, the presynaptic neuron synthesizing the neurotransmitter. So inside of the vesicles, there's a neurotransmitter. Okay, it's a chemical substance. So it packaging it in a multiple membrane enclosed compartment called as a synaptic vesicles. Okay, so what happened? So the first thing, when the action potential arrives at the end of the kenapa tak hujung hujung neuron tu, when before it, how it actually transport the action potential to the next neuron. Macam mana sebenarnya? This is what happened daripada hujung dendrite of the postsynaptic cell and the synapse of the presynaptic cell. So when the action potential arrives, is going to depolarize the presynaptic membrane. Ingat, ada the action potential, use the word depolarizing. Okay, and then when the action potential comes, depolarize, the depolarization is gonna, now dekat sini you are involving the calcium channel pula. Tadi along the axon only sodium and potassium kan. Ni dekat hujung ni ada calcium pula. So it's going to make sure the calcium channel open, allowing all the calcium ion to flow in, triggering an influx of the calcium ion. Next one, number 3, when the calcium ion is accumulated ataupun the concentration of calcium is really high over here, causing a synaptic vesicle to fuse, okay, with the presynaptic membrane. This is the presynaptic membrane. So the vesicles is going to slowly come over, fuse the membrane, and then release the neurotransmitter into the cleft over here, ataupun kita panggil gap junction. Okay. So, and then once it release dekat cleft, the synaptic cleft over here, is going to bind to the ligand-gated ion channel. There's a lot of ligand-gated channel over here in the postsynaptic membrane. Okay, this is the ligand lah. What does it mean by ligand-gated? Meaning it's going to allow the ion to flow in and out when there's a ligand binds to it. Ada ligand datang binds to it, only it's going to open. So the binding is going to trigger the opening, allowing the potassium and sodium to diffuse through. The same thing lah. 2 sodium flows out, 3 sodium flows in. Okay, we have also the generation of postsynaptic, what do you call it? Sorry, postsynaptic potential. Postsynaptic maknanya selepas tu. So the process of direct synaptic transmission involving the binding of neurotransmitter to the ligand-gated ion channel, normally we call as a ionotrophic receptor. So the neurotransmitter binding to the receptor, causing the ion channel to open, generates a postsynaptic potential, okay, which is a graded potential in the postsynaptic cells. So dekat postsynaptic potential, there are 2 different category. It's either excitatory postsynaptic potential ataupun kita panggil EPSP, where they're involving depolarization, where the membrane potential is going to move ataupun triggering to the threshold. Okay, and then we have the inhibitory postsynaptic potential, we call it as IPSP, where it's going to involve the hyperpolarization. The membrane potential is going to further away from the threshold. So after the release, the neurotransmitter is going to diffuse out from the synaptic cleft, may be taken up by the surrounding cells and degraded by enzyme. Maknanya bila the neurotransmitter is already taken up, yang mana yang selebih-lebih itu is going to be degraded by an enzyme. Dia akan hapuskan. And make sure there will be no any neurotransmitter left here over here. Okay, so this is the summation of postsynaptic potential. What is a summation? Is a postsynaptic potential combined together to produce even larger postsynaptic potential. So we have a sub-threshold. Okay, just a single EPSP. EPSP tadi tu yang ni tadi. The excitatory. Okay, so it's usually too small to trigger an action potential in a postsynaptic neuron. So you see, there are 2 different action potential at 2 different branch ataupun dendrite, but it is really too small, so dia tak sampai resting potential, you're not creating any dia tak sampai threshold, so they are not going to create an action potential. Itu kita bagi sub-threshold. We have the temporal summation over here, where you have a 2 excitatory EPSP to occur at the same synapse in the rapid succession. You see, over here there are 2 arrows, kan? They are happening at the same synapse. Okay, so it's when you're combining, when you combine 2 EPSP to, you can create an action potential because it's already reaches the threshold. Okay, and then we have the spatial summation, where the EPSP tu yang excitatory, produced nearly simultaneously by different synapse. 2 different arrow over here. You see, one red over here, another red over here, happening at 2 different dendrite. Okay, at the same time. On the same postsynaptic neuron adding together, you're able to create another action potential as well. Okay, and we have the spatial summation on IPSP and the EPSP. Okay, they produce nearly simultaneously at the synapse at the same region or in rapid succession, is going to have a larger hyperpolarization effect than comparing to the single IPSP. So over here sebab ada dua, one is excitatory, another one is the inhibitory. So even though there are two E1 and E2, okay, but because of ada inhibitory tu, you're not able to produce another action potential. So this is the one lah. Once you are releasing the neurotransmitter, everything has been binded to the ligand gated channel, and then allowing to trigger to transport the action potential to the next neuron. Tapi what happened to the extra ataupun the remaining neurotransmitter? After a response is triggered, the chemical cyanide return to the resting state. How it happens? By clearing the neurotransmitter molecules that from the synaptic cleft. You need to sapu it, you need to remove it. So how? Some neurotransmitter is going to be inactivated by the enzyme hydrolysis. So there will be enzyme floating around. They're going to eat, I mean hydrolyze ataupun digest the remaining neurotransmitter. Some of them is going to recapture into the presynaptic neuron. Dia masuk balik tempat asal tu because there's a channel over here. Meaning you can recycle the neurotransmitter. We don't want to waste it lah. So we just repack and then guna balik. Okay, so these are some of the neurotransmitter. Actually, you can read it lah. So we have the acetylcholine, most common neurotransmitter in both vertebrate and invertebrate. And then we have the amino acid, biogenic amines, neuropeptides, and also some of the gases such as nitric oxide and also the carbon monoxide. Okay. So let's see some of the the diseases and disorders regarding the nervous system. Okay, we have the mental, most of them is going to concerning on the mental illness ataupun neurological disorder. So for example, the schizophrenia. Okay, and then we have the depressive illness, which are the bipolar disorder and the major depression. Okay, and the major depression patient is going to have a persistent low mood. Okay, for the bipolar tu pula, they have manic and depressive phases. Tiba-tiba dia high mood and then suddenly they are going to be low mood. And we have the Alzheimer disease, okay, a mental ataupun brain deterioration characterized by the confusion or memory loss. Okay, we have a Parkinson disease as well. It's a motor neuron disorder causing by the death of dopamine secreting neuron in your substantia nigra. Okay, so this is your substantia nigra. So for the Parkinson patient, there will be dia punya substantia nigra tu tak nampak dah. Okay. So you have to remember there will be no cure for the Parkinson disease. Okay. All right, guys, thank you so much. I am going to continue with the muscular system on the next video. All right, thank you.